small t, then he
Right.
noticed that the offsprings are more dominant,
If, yeah, he
are
observed in
expressed
terms of, yeah, yeah. What, so what Mariam is trying to say, let me just quickly draw it here. So Mariam is talking about something called test cross, right, Mariam?
Yes, sir.
Parker? Yeah. So we discussed what is a test cross. A test cross is when you are not sure between these two alleles. So this will also be tall and this will also be tall. Like phenotypically, how will you differentiate between what type of tall plant you have? So if you take this one, which is homozygous tall, and cross it with a short plant, in the first, so this is parent. In the F1 generation, all progenies will be tall, correct? And everyone's genotype will be capital T, small t. How? How is this happening? Let's make a Punnett square to understand this. Capital T from one parent and small t from other parent. So all the progenies will be capital T, small t, capital T, small t, capital T, small t, right? So all will be tall. On the other hand, if you have this one, capital T and small t, so this is also tall, but when you will cross it with a short plant, and if this is the parent generation, you see that in F1, let's draw a Punnett square here. So capital T and small t from this parent and small t from this parent. So we see this will be capital T, small t, and these two will be homozygous recessive, correct? Make sense?
Yes, sir.
Yeah. So what it shows that in this case, these two will be tall, but these two will be dwarf. But in this case, all were tall. So if you do this cross and you figure out that all F1 progenies are tall, then you can say that the parent was homozygous tall. And if you do this cross, taking a tall parent and a dwarf parent, and you see that in the F1, half are tall and half are dwarf, 50% tall, 50% dwarf, then you can say that the tall parent was a heterozygous parent. Does that make sense, everyone?
Yes, sir.
Yes. So this is basically what Mariam is talking about. And when you do this cross, it is called a test cross because you are doing to test the genotype of the tall parent. Everyone is clear with this concept? Aisha, Rabia. So Rabia and Iram has joined us. Good evening. How are you both?
Good evening, sir. Good.
Good evening. Okay, great. So we were discussing what we did in the last class quickly. Parker was giving a recap. So she talked about test cross. Is that clear to all of you? Yes? Or is there any doubt? No? Okay. Yes. What else did we do, Parker, in the last class? Apart from this, did we do some? Yeah.
There are two types of crosses: test cross and a back cross.
Right.
Test is a type of back cross.
Yeah. Test is a type of back cross because we are crossing it back with a recessive parent only. This was the parent genotype to begin with. Okay, perfect. We also talked about some mathematical equations that describes Mendel's genotype and phenotype ratios that he found, right? Especially the
Yes.
genotype ratios. Yeah. Is that part clear to all of you? Is there any question about that part? Remember this AX plus BX whole square? And we can
Yes.
write it as half capital T plus half small t whole square. If you open it, ultimately you will get the ratio, which is like 1/4 capital T small t, sorry, 1/4 capital T plus 1/2 capital T small t plus 1/4 small t, right? And this is exactly the ratios which we get in the F2 generation of a monohybrid cross. Is that part clear, everyone? Any doubts? Perfect. Okay, so in today's class, what are we going to start with is, as I said, I ask you to go back and read linkage and recombination, right? So it's 14th June 23. So did you go back and read about linkage and recombination? Yes, no, people. How many of you read linkage? So what was linkage? How will you define linkage if I ask? I think we discussed this already. We talked about linkage as the hair. We talked about linkage as a exception to law of independent assortment. So anyone would like to talk about what was linkage quickly? Thomas Hunt Morgan found this in fruit flies. What was it? Aisha, would you like to explain this quickly? And if you don't see, if you don't remember, which is very, very okay to not remember things, you can just say that you don't remember and you would like it to be discussed again, and we will do that. Okay, so no judgments in my class of any kind. Okay, you are all allowed to forget, but then when we discuss again, you have to discuss with the same enthusiasm and intensity. Okay, am I clear, everyone?
Yes.
Yes. So Aisha, do you remember
Yeah,
what is linkage?
physical association of two genes.
Perfect. Physical. What do we mean by physical association? Are the genes holding hands together? What do we mean by physical association when it comes to genes? Logically, think logically. So what are genes? These are just the segments of DNA itself, right? So when can you say that two genes are linked together physically?
They are closely positioned along the chromosome.
Yes, that's a very good point. Perfect. And that is the answer. When two genes are very closely positioned, like when they're physically present close to each other on the chromosome, on the DNA thread, then they are called physically associated genes. And the chances of these genes to segregate and form recombinants is very low. So the higher the linkage, lower the recombination. Now in today's class, I'll tell you why and how this happens. But for that, we have to first understand the concept of, so linkage I can call as Physical, this was okay. Physical association, right? Between genes. Okay. Now, for that, Iram used the word chromosome. So how many of you can tell me how to draw a chromosome? So let's say I draw, I think I should use marker to draw it, I guess. Okay, let's try it. So you see something like this in your textbooks, right? Correct? And we call it a chromosome, correct? X-like structure. Okay, let's say these two are chromosomes. Now, in the center of the chromosome, this red dot that I'm making, what is this? This is centromere, right? And do you know what is present on the centromere? What is present? What is present at the centromere that keeps the two? So this, sorry? Kinetochore, yes. So centromere has the kinetochore, CHORE, which keeps the two chromatids together. Okay, so this is one chromatid. Let me draw it here or here. So this is one called one chromatid, and this is another chromatid. So in two chromosomes, when they are in this form, which means each chromosome has replicated already. So there are 4 chromatids, okay, when meiosis happens in these chromosomes. And let's say if this is chromosome number 1 and it comes from the mother, and this is also chromosome number 1 which comes from the father. So in this case, these two chromosomes will be called in this case, if both the chromosomes are same, they will be called a homologous chromosome pair. Is it clear? If one is chromosome 1 and the other is chromosome 2, we will not call it homologous chromosomes. We'll call it heterologous chromosomes. But if both the chromosomes are same, the only difference is that one comes from the mother and the other comes from the father, which happens inside our genome. Then the pair of chromosome is called homologous pair or a homologous chromosome. Is that part clear, everyone?
Yes, sir.
Okay. Also, you should remember that homologous chromosomes are exactly same to each other. They can carry different alleles, but the position of the allele, the number of genes that they carry, their length, the position of centromere and kinetochore, everything will be same. Okay, so write down about homologous chromosomes. Yes.
So these homologous chromosomes are also called as non-sister chromatids, right?
Non-sister?
Chromatids.
We'll talk about non-sister chromatids in a bit. Okay, chromatids are single. Okay, so this chromosome, what it can do is, let me use another color. Yes. So when it divides, what it can do is, or I can use black, white. So during meiosis, this just divides from the center. Can you see it? Like this. And separate. And this one becomes one chromatid and this becomes other chromatid. So every chromosome before meiosis, when it DNA replicates, it forms two chromatids. We'll come to chromatids in a bit, okay? But let's first focus on homologous chromosomes or heterologous. So this is a homologous chromosome pair. Now, suppose on this pair, there is a gene there's a gene present here for capital like tallness of the pea plant. Let's say it's a pea plant's chromosome. Then on the homologous pair also, this gene will be present exactly on the same location here. But it may or may not contain the same allele. If it is containing the same allele, then this plant will be capital T, and it will be homozygous tall. Make sense?
Yes, sir.
Yes, sir.
Okay. Just 1 second. Yeah. It is also possible that the other pair contains the other allele. In this case, what the plant will be?
Heterozygous tall.
Heterozygous tall. And if this one is small t, then
Homozygous.
homozygous dwarf. Perfect. Dwarf are always homozygous. So do you understood that the gene for height is present at the same position? If there is another gene for, let's say, flower color, let's say W, so it will exactly be same on the other one, and it can contain a different small w, different allele. But these positions where these genes are present are called locus of genes. So the locus, the position of the gene on homologous chromosomes is always same. And the number of genes that they carry is also same. And the position of centromere, write down these things. In a homologous chromosomes are similar to each other in carrying the same number of genes, at same locus, having the same position of centromere, and same length of their arms. So what do I mean by length of the arms is this. So you can, yeah. So this one is the shorter arm, right? Can you see that it is slightly shorter? And this one is the longer arm, right? Of the chromosome, make sense? So centromere is not exactly in the center. In some chromosomes, it is in the center. In some chromosomes, it is away from the center, on the basis of which we divide chromosome into different categories: telomeric, centromeric, etc. But we'll talk about it later. But you understood that if it is not in the center, it will have two different arms, right? One short and one long. The shorter arm is called P arm. The word P comes from petite. Petite means small in English. That's how you can remember P arm. P arm is always the shorter arm. And the other one is called Q arm. They didn't take much of an effort and just P and Q. Whatever came the next to P, they call it Q arm. So Q arm is longer and P arm is shorter. Is it clear, everyone?
Yes, sir.
Perfect. You know, this is how we actually define position of genes, the locus. So you will find sometimes, especially in in your higher studies, if you go on to pursue genetics, that it's written that this is gene X, let's say, and its position will be chromosome 2, P 113.4 centimorgan. Okay, it means that this gene is present on chromosome 2 on the P arm of chromosome 2 at this locus. And the distance that we measure is called centimorgan. It's not centimeter, it's centimorgan, named after Thomas Hunt Morgan. Is it clear?
Yes.
C is small and M is capital. So by looking at this, a geneticist can know that, okay, this gene is present on this locus, on this chromosome. Okay. And if it is Q, then you know it's present on the longer arm. Everyone clear with this concept? Homologous, heterologous is clear to you, people?
Yes, sir.
Yes, sir.
Perfect. Now, Iram brought about the point of sister chromatids and non-sister chromatids. So who are sisters? Who do you call your sister? The one who's born to your parents, right? Like in real biological terms, not in abstract friendship terms. But so if this is one chromatid, chromatid 1, this other chromatid is exactly like this, right? Make sense? So if this has capital T at this location, this will also have capital T. Now, this is not possible that one has capital T and the other chromatid of the same chromosome can have small t. This is not possible. Does that make sense? If here is W, here also it will be capital W only. Does that make sense? Similarly, this will be small t, and here will be small w. Is that clear, everyone?
Yes.
So these two chromatids will be called sister chromatids because they are exactly same to each other. They come from the same chromosome when it duplicates and forms two chromatids. That's why they are called sister chromatids. And if I take this one and this one, then these two will be non-sister chromatids. You know, non-sister chromatid can be this, and so any one chromatid from this chromosome and any one chromatid from this chromosome together will be non-sister chromatids. Okay, does that make sense, Iram?
Yes.
Yeah, so two different chromosomes, whether they are homologous or heterologous. We don't study heterologous chromosomes together because that makes no sense in genetics. Studying chromosome 1 with 3 is not of much use. So you study 1 with 1, like 1 from mother with the chromosome 1 from father. Okay. So sister, non-sister chromatids, everything, every concept is clear. Please quickly draw this so that you remember everything. And let me know when you are done.
Sir,
Yes.
sir, in the case chromosome, what is written in green?
Green, hair,
No, the other side.
hair.
Yes, sir.
This is shorter arm. Short. Oh, sorry, my handwriting is too bad. Let me write it clearly. Is it clear now? Shorter arm is called P arm and longer arm is called Q arm. Okay. Let me know in the chat when everyone is done. Just write that if you are done. I will wait for 1 minute for everyone to finish. A couple of minutes, yeah. Anyone has any doubts in between, just ask. Okay, so I think Aisha, are you done?
Yes, sir.
Okay, perfect. Waiting for you. Cool. So now that you understand the concept of homologous chromosomes, non-homologous chromosomes, let me quickly show you what. Let's see how we can understand recombination. Okay. So we know that for recombination to happen, there has to be chromosomal crossover. Okay. Now, what do we mean by crossover? So chromosomes can share genetic material with each other, just like we as friends share our, tiffin boxes, we share our things with each other. Chromosomes also can do that. And how do they do that? Is something like this. So let's say this is again one chromosome, okay? And this is the homologous pair of the chromosome, okay? To make things clear, let's also make centromere. Now let's say on this chromosome, we have two genes. You can see this blue I'm marking.
Yes, sir.
Capital T. The green here is, let's say, for color. And then there is one more gene, which is, let's say, here for what can be round and wrinkle. So let's say for round. So somehow this chromosome, which is from the mother, has all the dominant alleles. Let's just believe that. So this is homologous, so it will also have the same locus for the gene, but let's say somehow this has all the recessive alleles, small t, small w, and here as small r, correct? So if one chromatid has it, the other chromatid will also have the same, right? So what will be here? Keep telling me, Aisha, what should I mark here? Which allele will be present here, Aisha?
T.
Sorry?
T.
Sorry, I could not understand you.
Is it T?
T, of course it will be T. You have to tell me dominant or recessive, capital T or small t, because this is also T, this is also T. Yeah. Tell me. No, it can't be recessive because this chromatid is the exact copy of this. Do you see they are sister chromatids? They are attached with the same centromere. How can this be recessive? Because this is dominant. So where could this recessive have come from? The only way sister chromatids are formed is by replication, DNA replication. So one chromatid becomes two, but they are still attached with centromere. Is that clear? Look here. You drew this in your notebooks or not, Aisha?
Yes, I do.
So could you see here? Can you tell me what do you see here on chromosome 1 of both? On both,
T and capital W.
yeah, on both sister chromatids, same alleles are present. Okay, makes
Yes.
sense? Yeah. Till they are attached together with the centromere, on both the sister chromatids, you will find same alleles. So here also they will be same. It will be capital T here, and here it will be
Small t.
small t. What about this locus? Parker.
Small w,
Here,
capital W,
here capital W, and here
small w,
small w. Okay. Yes. And what about this locus?
capital R,
Okay. And
small r,
okay. So you now understand that these two are sister chromatids. These two are sister chromatids. These two are non-sister and these two are non-sister, right? These two also can be non-sister. And these two are also non-sister. You understand all the combinations? Yeah.
Yes, sir.
Okay. So this is how it is in the beginning. Now, what happens in crossing over is something like this. So we have to make the same chromosomes again. During crossing over, the two non-sister chromatids from the two chromosomes literally come together to form a association, like a physical association. Can you see here? This is called the crossing over. Oh my goodness. This is called the crossing over event. This here. Can you see this?
Yes,
They
sir.
physically cross each other. And because of this, let's again color code the alleles. And now quickly tell me what should be written where, which letter. So this will be capital T, small t, right? And this will be capital W, small w, and why is it so difficult to make a small w and small w? And then remember we here had capital R and we here had small r. But here, this arm and this arm gets very close, right? This is capital R and this is small r because it crossed over. Do you understand, everyone?
Yes, sir.
Yes.
Now this region is called the crossing over bridge, or we call it chiasmata. Okay, chiasmata or crossover bridge. Keep making this with me. First, you understand, I'll give you time to make it. Now, once this is formed, in the third step, where we will again make the chromosomes, we get, I think color coding it differently would have been a better idea. But since we made all the chromosomes black, let's keep it black. But what happens? This part goes here and this part goes here. There is an exchange of material, because of which the two chromosomes, the two homologous chromosomes will have these specific combinations. Okay, this will be capital T, small t, small t. This will be capital W here, small w and small w. But interesting thing is, here we have capital R and here we had small r. But now on this one, what we will have? Small r, and this will have capital R, you know. So this is how the exchange happened. You understood? Because of this chiasmata bridge. Okay. And now the next step, which is the final step in meiosis. In meiosis, you know that from one diploid cell, so this is a diploid cell, right? With 2 copies of each chromosome. Finally, we get 4 haploid cell. So it exactly happens through cutting off these, separating the chromatids. So finally, in each gamete, 1 chromatid goes. So what will happen is, you will have 1 chromatid and 2 chromatids which separated. This is the 3rd chromatid. And this will be the 4th chromatid. Okay, let's say. And they still will have, now they are detached from each other. And 1 chromatid will have the following combination. I'll first make all the colors of the locus. And then we'll talk about which allele they will carry. Yes, so this is chromatid 1. What will be the genotype? Let's tell me quickly. Capital T, capital W, sorry. Capital T, capital W, and
Capital R.
capital R. Okay, perfect. Tell me about this one. Second one. Let's say these two separated. Capital T, yes.
Capital W.
Capital W, yes.
Small r.
Small r, perfect. Then for this one.
Small
Third
t.
one, small t, okay.
Small w.
Small w, okay.
Capital R.
Capital R, okay. And for the last one, Aisha, will you like to tell for the last one?
Capital small t,
Small t,
small w,
small w,
and small r.
and small r. Perfect. Now, look here. These were the parents we started with, right? In parent, 1 chromosome had capital T, capital W, and capital R, and the other chromosome has small t, small w, small r. Now, when it formed these 4 chromatids in 4 different gametes, gametes are haploid. Now, again, when gametes will fuse, they will again become diploid and all. But you see here, can I say that, let's number them 1, 2, 3, and 4. So can I say that this 1 and 4 this one and this one are parental combinations, parental type? Because this was exactly what was present in parents. Make sense or not?
Yes, sir.
Yes.
But this one and this one, second and third, do you think these are parental?
Non-parent.
Yes, they are called non-parental combinations. Or there is another word for it. We call them,
Recombinant.
Recombinant.
yes, recombinants. And this is what I was talking about in the beginning, that if one parent has curly hair with blue eyes, the other has straight hair with black eyes, there could be progenies with curly hair and black eyes, right? Which is not like any of the parents. And now you exactly understand how does this happen. It happens through physical crossing over and chiasmata formation and sharing of. So the breakage, this breaking apart of chromosomes happen exactly at the same locus. Okay. It happens exactly here. An equal amount of information gets exchanged. So it's a very controlled exchange system. It's not like you can take more from one chromosome and can give less. If that happens, see, in biology is not foolproof. Life always tries to make itself better, but inside the living system, there are many things that can go wrong. And when they go wrong, then we call it disorders or diseases. When it happens in chromosomes, we call them genetic disorders or chromosomal disorders or diseases. Okay. If a bigger part got broken from one chromosome, but only a smaller part got exchanged from another chromosome. So you can see that one chromosome will have some additional genes which other chromosome will lack. Yes or no? Make sense?
Yes.
Yes.
That will be a chromosomal abnormality. Is that okay?
Yes, sir.
So out here, we see that out of these four, two are, parental combinations, and other two are recombinants or non-parental combination. But this is because I very simply and very conveniently took, just give me 1 second, people. Yeah. Yes, I very conveniently took all the dominant alleles on one side and all the recessive alleles on the other chromosome and only showed one crossing over. It is not always the rule that only one crossing over event will happen in one chromosome. There can be more than one crossing over. In fact, there are always more than one crossing over events that happen. So let's say a crossing over event also happened here, right? At this junction here. Then these major chunks will also get crossed over and you will find more recombinants. Do you understand what I'm saying? The more the number of crossing overs, the more recombinants you will get. Now, let me ask you a simple question. There are three alleles we have taken here, T, W, and R. So which of these three, which two alleles or which pair of alleles do you think is more linked and will show less recombination?
T and W.
T and W. Everyone agrees with that? Our conclusion of Parker, Aisha,
Yes,
and
sir.
Mariam? Yes or no? Rabia? Do you agree? What Iram is saying, that T and W will show less recombination because they are more linked. Do you agree what she is saying is correct? Yes, no?
Yes.
Yeah. And why is that? Because the sheer distance between these two genes is less, let's say around 2 centimorgan. But the distance between the W and the R is huge. And let's say, 20 centimorgan. So what happens is when you are randomly cutting, when randomly there is a crossing over event happening, if this crossing over happens here, still W and R will get separated. If it happens here, still here, 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 here. Anywhere it happens, the chances of W and R getting separated and making new recombinants is more. But the chances of W and T getting separated is very less, and it will only happen if crossing over happens in these locations. Yes? Do you agree? Less chance. The closer two genes are, the chances of they getting separated during crossing over events is lesser and lesser. And if two genes are just next to each other, let's say, then technically it's almost 100% linked, right? Or we can say 99.9% linked. Do you agree what I'm trying to say? Do you understand? So that's how some genes can form more recombinants, but other genes cannot form. So those genes which cannot form recombinants as much as they should are called linked genes. Now genes can have different degree of linkage. If we say that these two genes are 60% linked, we mean that 60% of the times the chances of they going together is there. And if we say that the genes are 90% linked, then we are saying that only 10% of the time they can form recombinants. So in this case, these two genes are linked. Okay, they show, or I should use the word, they show higher linkage, and hence they show lower recombination. And these two genes show lower linkage, and hence they will show higher recombination. Is that clear, everyone?
Yes.
Yeah. Would you like to draw this? Sorry, did I make it too small? Can you see this in one frame?
Yes, sir.
Yeah. Please draw this and let me know when you're done. Giving you a couple of minutes. I'm taking a quick water break, okay? Is that okay, everyone?
Yes, sir.
Yes.
Yeah. Okay. Thank you. Okay. Hello, everyone. I'm back. Everyone done? Yeah.
Yes, sir.
Okay, perfect. So let's just okay, good. So let's just do one thing. Now, since we understood recombination and crossing over, let me ask you one question. Do you remember the ratios from Mendel's crosses, specifically Mendel's Mendel's dihybrid cross? Let's take two genes, Mendel's dihybrid cross. Do you remember the ratios?
Yes.
Yeah. So what were the ratios? Let's go for the phenotypic and genotypic ratios. Okay. 1 second. Yes, I'm doing this exercise because I want to explain something which is in your NCERT, which is present there in a very, very complicated manner, but it's not that complicated actually. Okay, the phenotypic ratio of Mendel's dihybrid cross first. Anyone who remembers that quickly?
9:3:3:2.
Mendel's, let me just 1 second. Let me just write it here. Mendel's dihybrid cross phenotypic ratio. Okay, 9:3:3:1. Perfect. And what is the genotypic ratio? Come on, genotypic ratio. It's a slightly longer one, right?
So, I don't know
Yeah.
the order which you gave, but I can say.
Sorry.
So I don't know the order which you gave.
Okay. So it's always, I just remember it is in increasing and then decreasing like a pyramid. So it's 1:2 is to 1:2. So 1, 2, 1, 2 is to 4. I also gave you a trick to remember it. Is to 2 is to 1 is to 2 is to 1. It is a palindrome. No matter whichever way you read, it reads the same. If you read it this side, 1, 2, 1, 2, 4, 2, 1, 2, 1. If you read it this side, 1, 2, 1, 2, 4, 2, 1, 2, 1. Make sense? Iram? Makes sense?
Yes.
Do you remember? Yeah. But we are more importantly interested in the phenotypic ratio. Now, in the phenotypic ratio, can you tell me how many were, let's say, we are take any two genes, gene for height and color, okay, yellow. So how many were capital T, I mean, tall and yellow, and how many were short and yellow? How many will be tall and green, and how many will be dwarf and green? Which one is what?
9 will be
9.
tall and
So tall yellow is to tall
green.
green is to
Dwarf yellow.
dwarf yellow is to dwarf and green. We know that dwarf and green both are recessive characters and tall and yellow both are dominant characters. So how will I put this ratio here? What should be, how many will be tall and yellow?
9.
9. Okay. I don't know if it's right or wrong. You have to, someone has to just confirm also. How many will be tall and green? Actually, we don't need to know
3.
also. You'll figure out why. Okay. If you will give wrong numbers, we will catch it. How many do you think will be dwarf and yellow?
3.
Okay. And how many will be dwarf and green? 1, right?
Yes.
And what were the parents that we took? Now let's talk about parental combination. Parents that we crossed to get this was, we always start with pure parents, right? Yes or no?
Yes.
So parents could only be tall yellow, and we crossed it with dwarf green. Now, since you understand recombination now, tell me, which one are parental combinations? This and this, right?
Yes.
Can I say this is parental combination? And this one is also parental combination. And what about tall green and dwarf yellow? What are these two?
Recombinants.
Recombinants. Ta-da. This is what we are interested in. Now, according to Mendel, if anything follows Mendelian genetics normally, it should find this ratio in the F2 generation when we cross dihybrid traits. Yes or no? Yeah, you understood, everyone?
Yes.
Yes, sir.
This is exactly what the flies did not follow here, and because of which Thomas Hunt Morgan could discover linkage. Make sense now? This was the Mendelian ratio which the flies did not follow after he crossed a yellow body white-eyed with brown body red-eyed. Okay, so there was some issue, and the issue was that these two genes were not segregating independently. But if Mendel's third law is followed, then you should get this ratio in which total, how many progenies are total?
16.
16. So how many will be parental out of 16?
10.
Yes, so 10 out of 16 should be parental. How many recombinants?
6.
6 out of 16 should be recombinants. Now I just want you to find out the percentage. Tell me the percentage. 6 out of 16 is how many, what percentage? Sorry,
Yes, sir.
someone said something, but I could not understand. When you unmute yourself, just unmute and then take some time, 1 second, and then say
37.5.
37.5%.
5.
Okay. And what is the parental combination percentage?
62.5.
62.5%. So this is when normal Mendelian genetics is followed. Yes or no? Now, everyone, open your NCRT. Everyone has NCRT?
Yes, sir.
Yeah. Come to page number 84 quickly. I'll stop sharing for 1 second. Come to page number 84 of your NCRTs. Quick, quick, quick. Everyone
Sir,
came to, yes.
sir, can I rejoin?
Yes, yes, please rejoin. I'll wait for you. No worries.
So, which page number?
Page number 84. Just give me 1 second. I'm also sharing that. Can you all see the screen that I'm sharing again?
Yes, sir.
Yes, this page. Can you all come to page number 84? Can you all see this thing in your page number 84? I'm also, if some chapters have been deleted, I think the page number also will change in the new textbooks. This is from the old textbook, I'm sorry. But see this figure, it is in chapter 5. Come to this figure. You can all see the figure on your slides, right? And also tell me what is the page number in the new books after deleting some chapters. Why is no one speaking? Everyone found this figure or not? I'm waiting if you are.
Yes,
Found it, sir.
Okay, what is the page number?
84.
84.
84 only. Okay,
84.
perfect. Yeah, okay. Now look at this figure. Do you understand anything? First, let me tell you, these are two different things. You should see it. Cross B is separate from cross A. In cross A, they took two traits. Yellow body with white eyes and a wild type of it, which is brown body with red eye. Okay. In cross B, yes, Hiram, you
So
have a
wild
question.
here means dominant.
Sorry.
Wild here means dominant, right?
Wild, I means yes. Wild mostly is the in wild type, whatever expresses. Okay. So that's one way to write it. And wild can also be a mutated version, but here for the sake of simplicity, let's say if this is yellow and white, then this will be brown and red. So in this case, brown body and red eye, both are dominant characters. So just write it in your books only. I don't know why NCERT does these things to confuse. In the chapter they wrote, when Morgan, in their explanations, it's written Morgan crossed yellow-bodied, white-eyed flies with brown-bodied, red-eyed flies. But when they make the figure, they write yellow and white, but they write wild type. You could have just written brown and red. That makes life very easy. But maybe they want to teach you that wild type is where mostly dominant characters express, and then you make the homozygous recessive characters. So it's not like in nature you will not find yellow and white bodied flies. You will find, which means those are the recessive flies. Okay. So one cross was this, and about this we have already studied that when Mendel did this in F1 generation, either he got wild type or he got yellow and white. And when he crossed them together, he found in F2 generation that there were 98.7% parental and 1.3% recombinant. Then he did another cross, taking white-eyed and miniature body size. Like these were smaller flies, their size was small. So for body size also there is gene. Same is for humans. And a wild type, which mean red-eyed and bigger bodies. You can write here red and large, white and miniature, yellow and white, brown and red. Okay. When he took these two characters, he saw that in F2 there were 62.8% parental and 37.2% recombinant. Now tell me, on the basis of what you just calculated, which one follows Mendelian genetics and which one does not? Cross A or cross B? Look at cross A and cross B, and you already calculated some percentages above about recombinants and parental combinations. So which ones follow the normal Mendelian genetics, the law of independent assortment, and which cross does not? Aisha, you're going to tell me this.
Cross B follows Mendelian.
Cross B follows Mendelian genetics? How can, and why why do you say that? Mendelian inheritance is followed in cross B. Why do you say that? Because what you calculated, 37.5 should be the recombinants percentage if it's everything is happening according to Mendel's rule. And here it's 37.2, right? Almost same, right?
Yes.
But here recombinants are just 1.3%, which means what is happening in cross A here? What is happening here? What can we conclude from this? First thing, can we conclude that the genes, the two genes are linked? Yeah. There is around 99% linkage. Can I say that? Yes, no, something will help.
Yes.
Yeah. What else can I conclude? That because of linkage, there is very less recombination between these two genes. And pay attention to how they have drawn it. Did you get any hint? What else I can conclude from these figures? Can I conclude that the gene for yellow body color and white eye, which means the gene for body color and eye color, are located very close to each other on the chromosome? Can you see here? This is the distance. Whereas the gene for the white eye and body size are very far away from each other. Yeah. Can you see this?
Yes, sir.
Can we conclude this? Yes. So we can conclude that there is linkage because the genes are present very close to each other and hence there is very less recombination. So cross A is an exception of law of independent assortment and it does not follow Mendelian inheritance, whereas cross B follows Mendelian inheritance and therefore we get exactly the similar kind of recombinants in parental that Mendel also calculated we should get. Is that clear, everyone?
Yes, sir.
Yeah. Anyone has any doubts? Anything you want to ask? Oh. Okay, so today we will not start the next topic, which is pleiotropy, because it's long and we don't have time. So if you have any questions, we have a few minutes. Please ask me. Otherwise, that's where I will stop today at recombination. It's a very important topic. For sure, there will be questions from finding number of recombinants and progenies in your entrances. So prepare this topic with utmost, concentration. Also in your
So,
boards, there can be, yes.
so this experiment was done by T.H. Morgan or Alfred Stottler?
Sorry, this experiment was done by?
T.H. Morgan or Alfred Stottler?
What is the other name you are saying?
Alfred Stottler. There's one more name in NCERT.
No, this experiment where they figured out linkage was done by T.H. Morgan. Okay. I could not understand what was the other name. Can you spell the other name?
So I'll write in the chat.
Yeah, yes, please. Who is this person? Because even in your NCERT, if you see the legend of this figure, it's written that it's conducted by Morgan, isn't it? It should be, right? I'm not sure. Alfred Sturtevant. Okay, no. So Alfred Sturtevant, it's a very, how should I? He was an American geneticist who worked along the similar lines, but he was the one who first constructed the map, okay? The map for genes, the map where you measure the distances between, because Morgan did not know about their location of genes on the chromosome. So in, if I'm not wrong, around 1910 or 11 was the time where Alfred Sturtevant figured out the map for genes. Okay, that's called genetic mapping. So Alfred Sturtevant did genetic mapping for the first time. But this cross that they are showing here, cross A and cross B, was first done and figured out by Thomas Hunt Morgan, where linkage was discovered. Okay.
Yes, sir.
And Alfred Sturtevant, let me tell you this very interesting thing. He worked with Mendel. Okay. And Mendel was like kind of a, what do you say, advisor to Sturtevant, or colleague, I should say. The same relationship that kind of Watson and Crick had, you know. Watson and Crick, the famous duo who figured out double helical structure of DNA, right? You must be remembering. Similarly, once Morgan figured out linkage, then they started to ask more and more questions, and there came Sturtevant, and Sturtevant wanted to study what is the reason behind linkage. And then he figured out that chromosomes are located actually, sorry, genes are located actually close or far away from each other. That's so the genes which are located close to each other, they always show linkage. But the genes which are located far away from each other do not show linkage. So that was his contribution to make the genetic map. Okay, which is again a very, very important. Okay.
Yes, sir.
But this particular thing, I think, yes, you're right that they have also shown the distance here indirectly. This was not figured out by Morgan, but the crosses were done by Morgan. Yeah. Okay. Yeah. Cool. So let's stop here, and I'll see you all in the next class, where we will talk about different kind of genetic inheritance, pleiotropic and multiple gene inheritance, and then we'll go to some disorders. Okay. Pedigree analysis and disorders. Yeah. Pedigree analysis will be very interesting. I hope you will like it. Yeah. Okay.